Hi everyone! In this video, I will go over how an action potential is generated in a neuron. So first of all, let me go over some of the parts of a neuron. So in a neuron, there is a main cell body that I'm going to use a little shape like this to represent its cell body. Okay, so we have this portion of the neuron that is called the cell body. Within it, of course, there is nucleus, as well as other organelles that's within the neuron, such as rough ER, ribosomes, etc. I'm not going to draw all the cell body organelles because we're not talking about organelles of the cell here, but we are just mainly identifying all the little parts of main parts of the neuron. And what's coming out of the little appendages of the cell body in the neuron, these are what we call dendrites. Dendrites, remember, are for receiving signal. Okay, if we're talking about neurons, which are cells of the nervous system, remember they are for propagating electrical signal from one place to another, whether it be from the brain towards the periphery or towards other spinal nerves, etc. So it is dendrites that are receiving signal from a previous neuron or previous nervous system location, such as the brain. This location right here is what we call an axon, which is usually very long, cylindrical shape like that. And it can be as long as 1 meter, as depending on how long that neuron is and where is it connecting between. Some can be as long as 1 meter if they're, for example, if they got to go from your spinal cord all the way out to your thighs, for example. Then you have this section here. These little branches, they are called telodendria. And at the end here is where we have an axon terminal. Okay, axon terminal. All of these are axon terminals. There's multiples of them, but I'm just going to write one there to indicate that these are the axon terminal. This specific location right here, I'm going to kind of bracket it like that as a location before from the cell body and the axon. That location right there is what we call the axon hillock. Axon hillock. So what happens when we're trying to generate an action potential in a neuron? We have to receive signal from a previous neuron. So let's say this came from a previous neuron, and here is the particular axon terminal that is coming from what we call presynaptic neuron. If this previous one, if we could work to go on further and able to draw the rest of this will be entirely another neuron. That is what we call the presynaptic neuron. This one here then is what we call the postsynaptic neuron, because it is the neuron that is receiving the stimulus and the signal coming in. So now we have this particular axon terminal coming in from the presynaptic neuron that's going to stimulate the postsynaptic neuron to generate an action potential. How it does that is, remember that we will talk about this later too, that there are synaptic vesicles within axon terminals. And within synaptic vesicles, you have neurotransmitters. And neurotransmitters have to be released in a particular synapse area. So in this particular case, we're talking about a synapse that's between the axon terminal of the presynaptic neuron and the dendrites of the postsynaptic neuron. That synapse is very tiny. It's separated by a tiny synaptic cleft. And once the neurotransmitter is released into the synaptic cleft, it binds to particular channels, ion channels on the postsynaptic neuron side. And let's say in this case, we're talking about, since this is kind of tiny, I'll just draw it here. but it is in the synapse that these are sodium ion channels. Okay, sodium ion channels, and we would say that they are chemically gated. Chemically gated sodium ion channel because of the fact that it is these little neurotransmitter that can bind to these channels in order to open those particular channels. Now, once those channels are open, we'll come back to this concept once we get to, further down along the road. But for now, just bear with me and you will get to see again in a more enlarged version of what happens here. So in comes sodium ions that are coming in from the interstitial space. Sodium ion is not being released from the axon terminal of the presynaptic neuron. Remember, sodium ions are just floating around in the interstitial space around the entire neuron. So they're just flowing inwards from the channels into the cell body of the postsynaptic neuron. Now, all of these sodium ions coming in are going to collect at the axon hillock. So all the sodium ions are going to come here. They're going to float inside the cell body towards the axon hillock. What is the purpose of axon hillock is to collect all the charges so that as more and more sodium ions are being, absorbed into the neuron, they are coming into the neuron from the channels, they would increase what's called membrane potential. So with action potential in a neuron, usually we kind of depict what's happening in a neuron with a graph by the x-axis being time and the y-axis being millivolts. Millivolts is the voltage in which we use as a unit of measurement of membrane potential. So remember, this is your membrane potential graph here. So at resting membrane potential, okay, your neuron should be right about -70 millivolts. But as more and more sodium ions are coming in to the postsynaptic neuron, that will increase your membrane potential, but because these are positive charge ions. It will bring the membrane potential higher and higher. The more sodium ions coming in, the higher it will get. Eventually, it will reach a point where we get to -60 millivolts, which is what we call threshold. Once membrane potential of a neuron reaches -60 millivolts or the threshold, it will cause an action potential to be fired. So what's happening here is that when the membrane potential of a neuron reaches -60 at the axon hillock, it causes a membrane potential or action potential to be fired along the entire length of the axon in an all or nothing principle. Meaning that once it's fired, once the axon hillock has reached -60 as your millivolts of threshold, it will not turn back. Okay, it will just go on to the entire axon until it reaches the axon terminal. So threshold, that's why this is called thresholds, is because that's the point in which we're waiting for the membrane potential to reach to this particular millivolts before we can fire an action potential. Because let's say there's maybe 1 or 2 sodium ions, but it's not enough, and it just increased the membrane potential to -68, -65, but that's not enough to start a threshold. That is not where threshold is. So an action potential cannot be fired unless there's enough sodium ions accumulating at the axon hillock such that it fires an action potential when it reaches -60 millivolts. It can't be -62. It can't be -61. It has to be exactly a -60. Then what it will do is that you have all these. I'm just going to draw a few of them. But it does go down the entire length of the axon, like that. These are what we call your, I'm going to draw it right here, voltage gated sodium ion channel. Okay, remember it is voltage gated. Very important because it is very different between chemically gated and voltage gated. Chemically gated means that that channel will only open when it binds to a particular chemical, such as your neurotransmitter. Voltage gated means that it must be activated only by when the neuron's membrane potential have reached a certain point. In this case, for sodium ion channel, they will open at negative 60 millivolts and close at positive 30 millivolts. And that's just the way it is. You might, well, why is it that that's the case? Well, that's just the way those voltage gated channels are for sodium ion channels, is that they open at negative 60 and they will close at positive 30. So when your membrane potential at the axon hillock has reached negative 60, it causes all of these sodium ion channels to open. When they open at the beginning of the action potential, there should be a lot of sodium ions on the outside. Hence, all of them will rush in towards inside of the neuron. So when sodium ions are now rushing in very rapidly into the axon, and it will then bring up the membrane potential to higher, higher, and higher, because you're putting more positive ions into the neuron itself. Now, what drives these sodium ions to go into the neuron is a result of electrochemical gradient. I'm not going to explain that here. I might explain that in another video. But remember that inside of the neuron is negative charge, whereas outside of the neuron is positive charge. So that is one of the key things that drives sodium ions towards the inside of the cell when the channels are open, because there's an attraction of charges. In addition, there's also a concentration gradient where sodium ions are more on the outside compared to the inside. And we know that, concentration gradient rule is that things always move from a higher concentration to an area of lower concentration. So this will increase its membrane potential as more and more sodiums are being going in to the neuron through these channels and reach a positive 30 millivolts. Now, once they reach positive 30 millivolts, sodium ions channels will now close, close up. So imagine that means that is closing up. And it will activate a different set of voltage gated channels. And I'm just going to draw it in between. Okay. And those are, I'm just drawing 3 for now, so I have room for the last thing here. But then you know that there are equal number of sodium and potassium channels. So this one right here is what we call voltage gated potassium channel. And for a potassium channel, they open at positive 30 millivolts and close at negative 70 millivolts. So when membrane potential at this point right here has reached to positive 30 up here, a maximum where the neuron membrane potential can go, all the sodium ion channels are now closed. On the other hand, voltage gated potassium channels will now be open. Once it's open, it actually drives potassium ions to move from inside of the cell to outside of the cell, because there are a lot of potassium ions inside the neuron. Again, due to electrochemical gradient, it drives potassium ions outwards outwards from the neuron. Okay, so remember, when an action potential occurs, sodium ions go inwards into the neuron. Potassium ions will go outwards out of the neuron. And because now potassium ions as a positive charge ion is moving outwards from the neuron, the membrane potential will now move downwards, become more negative again. Because of positive ions coming out, membrane potential will become more negative. And the fact that, there is something where the potassium channels are actually slow channels compared to sodium channels. So even though it is at -70 and reach that, it only begins to close at -70. And then it keeps going downwards because it overshoots a little bit to -90 millivolts due to the fact that potassium channels closes very slowly. And this overshooting of the membrane potential is what we call, I'm gonna use a different color, hyperpolarization. Hyperpolarization, where membrane potential becomes lower than the resting. Okay, so we have resting potential reaches -60, we call that threshold. And when membrane potential climbs to a +30, right here, this whole climbing process is what we call depolarization of the neuron. On here, on the other side, when we are at the maximum, climbing downwards to negative membrane potential is what we call repolarization. Okay, repolarization. Now, eventually, though, your membrane potential will climb back up to resting, and we're back at -70 at the end of the day here. So let me put the little dots for -70 resting, -60, +30, -90 where it overshoots a little bit, and then you're back at -70. -7, remember, is resting membrane potential. Now, what allows this to climb back up to resting is all due to a very important enzyme called the sodium-potassium pump, or you may have also heard it being called as sodium-potassium ATPase. So what this particular enzyme does is that it allows sodium ions to move outwards and allows potassium ions to move inwards, and it does so in the unequal distribution of sodium and potassium ions. This, let me again label that as your sodium potassium pump for this particular guy right here, and it will move 3 sodium ions outwards and 2 potassium ions inwards, which, you can also say it as for every 3 sodium ions that it move out of the neuron, it will always move 2 potassium ions into the neuron. So the sodium potassium pump does 2 things. Let me write it on the side here. Sodium potassium pump does 2 things. 1, reset the neuron, where it will allow for sodium ions to be on the outside and potassium ion to be on the inside. Okay, it reverses the ions back to reset the neurons so you're ready for the next round of action potential. Because what happens is when you don't have enough sodium ions on the outside, you can no longer allow them to come in for depolarization to happen. In order for this particular neuron to fire another action potential, you must reset the ions back to their specific location. So after sodium ions have come in through the action potential, they have to go back out in order for another action potential to be fired. So this is why you need the sodium potassium pump here to reset the neuron, put all the sodium ions back on the outside, and put all the potassium ions back on the inside. Second thing, it will bring the neuron's membrane potential back to -70 millivolts. -70 millivolts, which means it's back to resting membrane potential. Now, that is really pretty much what an action potential does in a neuron, what fires it. What do we mean by depolarization, repolarization, and hyperpolarization? What sets off the action potential down the length of the neuron? It's all due to all the ions collecting at the axon hillock. So now what happens when an action potential has been fired down the axon towards the axon terminal? I will then talk about that next.